So we'll go ahead and get started. First, wanted to thank everyone for joining this webinar. This series is designed to orient new clinical research sites and then their staff to the HIV Prevention Trials Network. Now, this series is going to consist of 2 webinars, and each webinar is going to be an hour long. This webinar is going to provide a general overview of the Leadership and Operations Center. The second webinar is going to focus on the Statistical and Data Management Center, and then also the HPTN's Laboratory Center. And this webinar is going to happen on October the 11th at this same time. So if you haven't already registered for that one, please consider registering for that webinar too. Just a couple of logistical things. I wanted to note that this webinar is being recorded. The recording is going to be posted on the HPTN's website, www.hptn.org, at the end of this webinar. And this webinar is going to be available to anyone at our sites who would like to access it in the future. If you hire new persons in the future and you'd like to use this as part of your orientation packet, we would encourage you to do that. All lines for this call, they have been muted. If you do have questions for the speakers, please type those questions in the chat box. Also, at the end of the presentation, the webinar, there's going to be a brief survey. If you could please complete that survey, as the survey helps us to plan for webinars in the future. That information that you provide to us is quite helpful. With that, I'm going to introduce our 2 speakers who are going to be leading today's discussion. Mrs. Kathy Henson and Dr. Nirupama Sista. Now, Kathy Henson, she's the HPTN's Associate Director for Finance and Administration. She provides programmatic, managerial, and administrative support for the HPTN. She's responsible for the financial management of the HPTN's Leadership and Operations Center agreement. She establishes and monitors contractual agreement for protocol implementation funds and financial management of sub-agreements. And she also provides clinical trial units budget guidance, and she reviews and develops protocol-specific budget templates. Dr. Nirupama Sista is the director for the HPTN. She leads the development and implementation of all ongoing HPTN research studies. She also focuses on strategic partnerships with funding agencies and collaborative partners. Dr. Sista is a senior research scientist with FHI 360, where the HPTN's Leadership and Operations Center is housed. And she has more than 20 years of experience in academic, pharmaceutical, and nonprofit sectors. Before joining FHI 360, Dr. Sista was a senior clinical research scientist for Triangle Pharmaceuticals, which is now Gilead Sciences. She earned her PhD in microbiology from the University of Tennessee with a postdoctoral training at the University of North Carolina at Chapel Hill. With that, I will turn it over to Dr. Nirupama Sista, who will get us started.
Thank you, Jonathan. Good morning, everyone. I'd like to extend my welcome to the staff from all the new and old sites. Welcome to the HPTN. So as Jonathan mentioned, this presentation is really geared for those site staff that are either unaware or want to refresh some of the structures and processes of the HPTN, and that includes 3 central resources: the Leadership and Operations Center, the Statistical and Data Management Center, and the Laboratory Center. So if I can have the next slide. And so the outline of my talk is going to be what are we, where are we, and who are we? And I hope that I can clarify or explain what and how HPTN functions through these set of slides. And I'll be joined in this presentation, as Jonathan said, by Kathy Hinson as well as Jonathan himself. Next slide. So, as you all know, the HPTN is really a collaboration between all of us, both all the sites, the staff from the sites, ethicists, community and its community participants, and other partners. And our main aim is to develop and test the safety and efficacy of interventions designed to prevent acquisition of HIV and also transmission of HIV. Next slide. And the mission of HPTN, particularly in this iteration of this network, is to advance the prevention research by developing integrated strategies, by combining different prevention interventions, and then in particular, improving systemic pre-exposure prophylaxis by evaluating new agents for systemic pre-exposure prophylaxis. Okay, I'm going to pause there, and I'm not quite sure what's going on my slide set. I don't know if you all can see. Should I do this? Or?
Yeah, hold on. Yeah.
Just a second. So, hold on one second. I think there is an issue here, but I'm going to go ahead and continue, assuming you all can see the set of slides. Can you see the set of slides?
We can still see the Nero.
Okay. So I'm not quite sure what's happening on this end, but I'll just go ahead and continue with it. So if I'm slide on,
keep going.
I'm on slide 4, and that just presented to you what our mission is. So the next slide shows you our research legacy, and I'm going to say next slide just for all of you to know, because I have somehow lost control over here and I don't see the slides that you are seeing. But I think what you should be seeing is a slide on our research legacy, and this is really to remind everyone that the investigators involved in HIV Prevention Trials Network have been involved in doing research in HIV prevention for more than 2 decades. And the very inception of HPTN started out back in 1993 as a international master contract called HIVNet. And the slide shows you the portfolio of that network or that contract, and Ward Cates and Steve Self were the principal investigators in that iteration of HPTN or HIVNet. We then moved into 1999 and the first round of HPTN with a portfolio that included microbicides, mother-to-child transmission, as well as antiretroviral therapy for prevention and other interventions. In 2006, Sten Vermund and Dr. Sukaresh Abdul Karim became the principal investigators, and then the focus continued more on antiretroviral therapy, STI treatment, substance use, and behavioral and structural interventions. And now, starting in 2013 through 2020, we are in what we call HPTN 3. Doctors Wafa El-Sadr and Mike Cohen are the principal investigators of our network. Our focus for this iteration is the integrated strategies, as I mentioned, and that includes PrEP with a particular focus of evaluating new systemic PrEP agents. So over the years, and this includes all these iterations, HPTN has conducted or continues to conduct or have completed 50 trials, more than 50 trials, all over the world. Over 50,000 study participants have been followed through our network, and this has resulted in more than 300 publications in peer-reviewed journals. If I can have the next slide. In this iteration of our network, we have been leaders in integrating strategies for prevention, and those strategies have included biomedical interventions such as Truvada for PrEP or injectable cabotegravir that we are going to evaluate as a systemic agent, behavioral interventions, and we've also conducted studies in structural interventions. Next slide. And our approach for how the scientific agenda is set, it is focused on both the HIV-infected participant in order to prevent transmission from the HIV-infected participant to an HIV-negative partner, as well as preventing acquisition in an HIV-uninfected participant. And antiretroviral therapy for the HIV-infected participant is the anchor for our studies where we focus on the HIV-infected participant. In addition, integrating that anchor with behavioral structural strategies as well. Similarly, with the HIV-uninfected participants, PrEP becomes the anchor, again combined with behavioral and structural agents where possible. Our approach is to evaluate new interventions or new approaches or feasibility of doing studies in new populations in smaller studies first, called Vanguard studies, and that then leads into larger studies, which ultimately has public health impact. And the next slide really shows a very successful history of doing just that. On this slide, there are many studies, the results of many studies that are highlighted, but I just want to remind everyone that the HPTN 052 study, which was named the 2011 Breakthrough of the Year by Science magazine, demonstrated that treatment of the HIV-infected partner reduced transmission by 96% in his or her negative partner in a study conducted in serodiscordant couples. And this study, these set of studies and all of this work cannot be done without the staff and participants at our sites, and there are more than 90 sites in more than 15 countries that are participating in HPTN research. And the next two set of slides shows you where our studies are being conducted. We have on here some of the sites that are also conducting studies that are in collaboration with our sister network, HIV Vaccine Trials Network. For instance, the site in Switzerland is involved in our AMP study, which is a collaborative study between HPTN and HVTN. And we have many sites in Africa that are conducting HPTN research or research in collaboration with HPTN and other networks. Our leadership, as I mentioned earlier, consists of Dr. Mike Cohen at the University of North Carolina and Dr. Rafael Sader at Columbia University, and in consultation with the National Institute of Health and the Executive Committee, the leadership sets our scientific agenda. The Executive Committee is shown on the next slide, and this is just a schematic, and I'll go into a little bit more detail in the next couple of slides, but it consists of the PIs of our network, international lead investigator, the past PI and co-PI of our network, as well as the PI, the director of our central resources, representatives from the community, from NIH, and sites. We also have several oversight committees, as well as scientific committees and working groups. Next slide. So our network structure consists of our executive committee, and really on this slide are listed the individuals who are the voting members of our executive committee. We also have many non-voting members that contribute to the science and governance of our network. Next slide. We have several oversight committees, our protocols, and I will allude to this in the next set of slides as well. Our protocols are reviewed by a committee, a subset that is delegated by our executive committee, and that committee is called as the Science Review Committee. It is chaired by Dr. Sten Vermund. All our studies are monitored on a regular basis for accrual, for retention, for study conduct, adherence to protocol. by our Study Monitoring Committee, and that is chaired by Dr. Jim Hughes. All the manuscripts in HPTN that are generated by investigators, protocol chairs, any manuscript that uses HPTN data gets reviewed by the Manuscript Review Committee chaired by Tom Coates. And the evaluation of sites in our network is conducted by our Performance Evaluation Committee chaired by Dr. Abdul Karim. Our scientific committees are geared to ensure that our focus remains on the populations at risk. And so, as you see on your on the slide, we have adolescents at risk, women at risk, substance users, men who have sex with men, and then, of course, integrating all our strategies in all populations. So, in order to maintain focus, our scientific committees continue to evaluate new science as well as propose new concepts. And we also have working groups. The participants are well represented with our chairs of our community working group, Melissa Turner and Nintendo Yola. I think we are probably the one group that has an ethics working group that ensures that all the studies that we are conducting in the network are ethically sound, and that is being chaired by Jeremy Sugarman and Jerome Singh. And in order to standardize as well as inform our science, we have a behavioral science working group chaired by Dr. Nabila El-Bassel. Next slide. So the network structure in depth consists of 3 central resources: Leadership and Operations Center here at FHI 360 in Durham, North Carolina, the Statistical and Data Management Center at SHARP at the Fred Hutchinson Cancer Research Center in Seattle, Washington, led by Deborah Donnell, and the Laboratory Center at Johns Hopkins University in Baltimore, Maryland, led by Sue Eshelman. And very briefly, our Statistical Data and Data Management Center facilitates all aspects of data collection, reporting, and statistical analysis. And you'll hear a lot more about this in the next session of this series. The Laboratory Center collects, tests, and reports results from biological specimens, assists in the development and quality assessment of local labs, and identifies and implements state-of-the-art assays. And again, you will hear more about them in the next session. So now I'm going to focus primarily on the Leadership and Operations Center. As I said earlier, we are located at FHI 360 in Durham, North Carolina, and we also have staff in our Washington, D.C. office as well as other parts of the United States. We are, I'm sorry, can I stay in that? We are involved in our clinical research and operations. We facilitate and we help develop our concepts. We are also responsible for the communications for the network, community engagement, and as Kathy Hinson will present to you, the finance of our network as well as the governance of our network. Next slide. So our clinical research managers, many of you have already interacted with many of them. They along with the research scientists, research assistants and scientists, really are the focus of the new research ideas that get developed. They are facilitators and they shepherd these concepts through protocol development, through activation of sites, protocol implementation, all the way provide oversight as the study is being conducted, and ultimately with a successfully completed study, manage the publication of results and dissemination. Next slide. Our communications staff consists of communication officers, web managers, and data managers. They manage our digital and social media platform, all the alias lists and directories that some of you have already been part of. They maintain our network database. They plan for all the study-specific communications as well as information dissemination, help design study-specific as well as HPTN graphics, including logos and templates, and they manage the logistics and planning of webinars as well as the large meetings that we conduct annually in Washington, D.C., and as many of you know, we are going to have our regional meeting 18th through the 20th of October in Johannesburg, South Africa. Next slide. I'm going to just transition to Jonathan, who will speak about the community engagement, and then who will then transition to Kathy Hinson, who will talk about the financing of our network.
Thank you, Dr. Sista. So the Community Involvement Program, we oversee issues that are related to community involvement and participation in the HIV Prevention Trials Network. As a group, we work directly with clinical research site staff and then also community advisory groups at those sites. We work with other HPTN teams and committees to help coordinate mutually beneficial partnerships with our community stakeholders throughout all levels of the HPTN's research efforts, with an ultimate goal of strengthening our already robust scientific portfolio. I want to take just a little time to highlight some of our roles and responsibilities, as I think that will be helpful in providing just a little bit of insight into what our day-to-day work is like. So the Network Community Involvement Program staff, we support the HPTN's Community Working Group, and this group is comprised of one community educator and one community advisory group representative from each of the clinical research sites that's conducting research in the HPTN. And as Neeru spoke about earlier, this group provides guidance to the network leadership and has representatives on all of our scientific committees. Along with the HPTN Community Working Group, the Community Involvement Program staff, we advocate for community involvement in the HPTN infrastructure and then also with other HIV research community programs. The Community Involvement Program staff, we assess community engagement and education activities and the advisory processes through annual community work plans that the site completes. We do routine conference calls, and we also make visits to the community advisory groups and community education staff at clinical research sites when such visits are needed. We'll assess and respond to Leadership and Operations Center identified and also site identified technical assistance needs by providing appropriate guidance and training needed to conduct HIV prevention trials in a community context. And we'll do this for our clinical research sites and also their community advisory groups. We also advise those clinical research site staff on community involvement activities, integrating those activities in the research process at the site level, the network level, and then we also help sites with community planning and then also budgeting issues. Now with that, I'm going to turn it over to Kathy Hinson, who is the fiscal director for the HPTN.
And thanks, Jonathan, and thanks to everyone who is on the call today. We appreciate your making the effort to be with us. You know, I'm here representing a team of at least 4 other individuals from FHI 360, many of whom you have received emails from for either requests or as follow-up. But the team of Sarah Stone, Gloria Faribault, Phil Hoover, and Beth Horn also helps support the fiscal program that's located here at FHI 360 to support the HPTN. Overall, our goal is to manage both our internal leadership and operations budget, but primarily to help manage the site's protocol funding. And as I mentioned, with a staff of 4 and 5 people, you know, we have experience in both protocol costing, grants management, accounting, auditing, and compliance, and so we do serve as a resource to the network. As many of you know, that's kind of who the team is and what our goal is. But as many of you know, the networks overall are funded by the National Institutes of Allergy and Infectious Diseases through a cooperative agreement, but that also includes funds from NIDA, other institutes within NIH, including NIDA, NIMH, and we've been successful in also working with pharmaceutical companies, including ViiV and others, as well as the Bill and Melinda Gates Foundation, to secure funding on behalf of the network. It is a 7-year award that began in January, as Niru alluded to earlier and showed the legacy of this network. And it is currently, similar to what many of you already know, the current award goes through November of 2020. The fiscal program, as I mentioned, includes all the institutions that went through this peer review process, as well as some specific protocol-specific sites that we've included over the years to carry out the mission of the HPTN. When I refer to funding, you've often heard the word core funding, and I'll go into this in a little more detail, but that's the basic infrastructure, core management and infrastructure funds that are awarded from NIAID, along with the protocol implementation or protocol-specific funds that are awarded from FHI 360. So on this schematic is really to try to show how the funds flow. People always kind of want to know where does the core come from and where do our protocol-specific funds come from. And this is not a simple schematic, but I think the main take-home message from this is that NIAID, Grants Management and Program work together to make a notice of award with core dollars to the clinical trial units. In addition, FHI 360, as the Leadership and Operations Center, receives the protocol funds in our notice of award, which again we then give to the clinical trial unit, and many of you who either represent a clinical trial unit or clinical research site are familiar with this process, but the protocol funds go from the LOC to the CTU or clinical trial unit and then are awarded to the clinical research sites. As I mentioned, there are some protocol-specific sites that we have brought on, and as a result of that, FHI, as the Operations Center, is directly providing the funds to that CRS. This is not a schematic, but it basically tries to portray the same message that the funds are at a site level are coming from NIAID directly, and that our protocol funds are coming from FHI 360 to the sites. And those protocol funds can look anything like development funds as well as implementation funds. And they may come in different ways. One include in terms of how payments are made, whether they're cost reimbursement or what we've tried to evolve into a per participant milestone-driven reimbursement. So again, I mentioned the funds go to the clinical trial unit, and that is different than many of the other networks. And again, that decision was made early on in the HPTN to get greater flexibility to shift resources as needed, to improve our tracking and strengthen our relationships with the sites, as well as monitor and be able to report expenditures. Now, I won't necessarily go into these NIH regulations that govern the cooperative agreements, because those sites that are both clinical trial units and clinical research sites are familiar with many of the policies that govern our funding. But just, again, I think the main message here is that there is an NIH grants policy statement that does, we are all compliant to those rules and regulations set out by the U.S. government. as part of that, obviously part of our compliance to ensure that we continue to receive funding is our audit requirements and compliance. I think because many of you may be involved in the reporting of expenditures to NIH, I just wanted to highlight, as you're familiar, each year we are required to submit an annual report, which includes the budgets for the following year. And again, thanks to many of your efforts, we were able to do that successfully for our next funding period. Those budgets and the deliverable is called an RPPR. It's a report that is done electronically, but it is submitted October 1st to receive funding for December through November of the following year. And as any program, when you receive funding, you always have to report on how you spend it. And this is just a highlight of some of the key reporting that we do, both internally within our network, but also to our sponsors at NIAID and other sponsors. So I try to highlight just a few things about the fiscal program, not being very specific whatsoever, as we try to get on protocol calls to highlight anything specific to how a protocol is actually being funded. But I welcome at the end any additional questions or offline discussions with people should they have any questions specifically about their site or their CTU. But I want to transition back to Niru to highlight the governance function within the LOC.
Thank you, Kathy. So the next slide that you all should see is about the network governance, as Kathy mentioned, and we wanted to just highlight that the Leadership and Operations Center here at FHI 360 convenes the executive committee as well as other meetings with other committees. There is an international scholars program that HPTN hosts, and many scholars have been recipients of awards from the HPTN International as well as domestic, actually, scholars program. There is a equally and in fact more robust domestic scholars program that has been ongoing for several years. Erica Hamilton, who is at the Leadership and Operations Center, is the coordinator of both the domestic and the international scholars program, and that is also housed in the Leadership and Operations Center here at FHI 360. As I mentioned, we conduct the network evaluation. It is chaired by Dr. Qureisha Abdul Kareem, but again, the network evaluation coordinator is at the LOC. Her name is Anna Levier, as well as we have members on the committee from our other central resources. The LOC organizes network-wide meetings, the annual meeting, the regional meeting, any other stakeholder meetings, any other kind of meetings, including protocol meetings. And then, as Kathy mentioned earlier, we produce regular reports that report back about our study conduct or performance to the NIAID Division of AIDS as well as to our leadership. I'm going to transition into some of the HPTN processes as part of our network governance. We also manage the processes within the network. I wanted to highlight that the manual of operations is on our website, the website that Jonathan alluded to, www.hptn.org. This is really a very good resource for anyone who wants to know anything about HPTN. It has many chapters, so starting from our leadership and description of our central resources and their scope of work, all the way through how publications are managed and generated in the network. So I would, again, encourage everyone to take advantage of this resource that is easily available on our HPTN website. Very briefly, I wanted to talk about our HPTN concept and protocol development and site selection. So the slide is really captures a very an overall capture of what happens during protocol development and execution, and then I'll go into a little bit detail with respect to the protocol development. But concepts come into our HPTN and are reviewed and approved or rejected by the HPTN Executive Committee, and they then give us the signal to develop the concepts that are approved into protocol, which then gets reviewed and approved, and I'll talk to you about it in the next slide. All our sites then get the final approved version 1 protocol that gets reviewed by the regulatory agencies in country. If they are international sites, some of the studies are actually under IND, and therefore the protocols get reviewed by the FDA in the United States. And once we have all the approvals in place and the sites are ready, the sites get activated for protocol initiation. And as I mentioned, the LOC is involved in the oversight of the study as the study is ongoing. Our study managers or clinical research managers also conduct regular site assessment visits. They conduct the training for the protocol to begin with, and then continue with assessment visits throughout the study. And this, I wanted to just highlight, is different from the monitoring visits that are conducted by the Division of AIDS assigned clinical research monitors. And as I mentioned earlier, the staff are also involved in results dissemination. So this is a schematic of our science generation process. Ideas can be generated by investigators or our scientific committee members, as I mentioned earlier, or are often centrally developed based on the direction that is provided by our sponsors or funders or our leadership. And those concepts then get submitted to the HPTN EC for review. If they are rejected, they go back to the authors of the concept. If they are approved, they then continue through protocol development with a schema that is finalized by the protocol team, and then ultimately a full protocol that gets reviewed by the HPTN Science Review Committee that I mentioned earlier, chaired by Dr. Sten Vermund, and approved by the SRC. Once we have a protocol that the HPTN has approved, it then moves through the Division of AIDS research review processes, including the review and approval from the Prevention Science Review Committee, all the regulatory reviews and approvals of dates, the medical or program officer review, and ultimately the regulatory affairs branch of the Division of AIDS reviews it, and it's signed off by the chief of the regulatory affairs branch. That version then is called version 1, which is disseminated to the sites for their reviews. Again, all of this is highlighted in great detail in Section 9 of the Manual of Operations. I'm going to touch very briefly on study site selection. I know many of you are probably interested in our process and how does that happen. Once a concept is approved for protocol development, and as the protocol is being developed, a site questionnaire gets developed by the study team members from the central resources to assess capacity at the site specific to that protocol's requirement. A site selection committee, which is designated and delegated by the protocol team and the leadership, discusses completed questionnaires that the sites provide back. Once we send out a questionnaire, and many of you have received those sort of questionnaires, you all respond to them, and those then get discussed and reviewed, and based on the requirements of the protocol, sites get selected. Sometimes, to be able to make final decisions, there are some pre-site selection assessment visits, often to evaluate infrastructure or capacity, or if it's an entirely new site, to ensure that site can get activated in a reasonable time frame and meet all the requirements of the Division of AIDS as well as of the network. Those sites that are selected by the site selection committee, then the list gets recommended to the HPTN EC. the HPTN votes and recommends it to NIH, and ultimately those sites that are approved by the EC and the NIH are considered for the protocol. Our questionnaires, again, very quickly, you all have received some of them, but the examples in our questionnaires are about HIV prevalence and incidence, experience in performing similar clinical studies. And I'll pause there and explain why we ask these from a study-specific questionnaires. As you all are probably aware, those of you who are involved in HPTN research, not all of our studies are identical to each other. And many of them are not even in the same populations. We do engage and enroll participants and have studies in people who inject drugs, in men who have sex with men, in adolescents, in women who are at risk in Africa, and in the general population, as well as in communities, where we have some community randomized trials. So because of the unique needs of our protocols, these questionnaires are catered to those protocols and then evaluate a lot of the criteria. Some of them are listed on this slide. And the process of prioritization after we receive these responses are really guided by the policies of the Division of AIDS. And we first look at HPTN approved clinical research sites, those that are already affiliated to HPTN. If we do not have the sites there because of either their overcapacity or needs of the protocol, we then go out to the other network approved clinical research sites. And if we still need to expand further, then those clinical research sites that are affiliated to the CTUs, but they were not perhaps selected in the last competition. And then as a final option, we go to totally new non-network sites. And when do we do that? We do that when we need unique populations. And for example, for people who inject drugs, we went into sites in Ukraine and in Indonesia and in Vietnam. We've done, as I indicated, we've conducted and are conducting the largest community randomized trial, a HPTN 071 study or a POPAT study, and we go out and look at communities uniquely needed for that study. And again, under specific settings, as we did with a study in the United States called HPTN 065. And we identify these non-network new sites based on availability of relevant epidemiologic data, whether research has been conducted by the site relevant to what is being proposed in the study, and of course the experience and expertise of site researchers, infrastructure, as well as the ability, as I mentioned earlier, to meet dates and HPTN requirements in a reasonable time frame. The site selection committee is chaired often by the protocol chair unless they have a conflict of interest, and it also consists of representatives from the central resources and DAIDS. And once again, I want to highlight to you that this is all outlined in the Manual of Operations in Chapter 20. Of course, all of our work cannot be completed at all without the National Institute of Allergy and Infectious Diseases, who are the funders of our network, and many of our studies are also funded by National Institute of Mental Health and the National Institute on Drug Abuse, all of whom are part of National Institutes of Health. And as Kathy mentioned, we also have other funders such as the Bill and Melinda Gates Foundation, OGAC, PEPFAR, as well as ViiV Healthcare. And I think that really sums up what HPTN is. We all will be happy to take any questions.
Thank you, Neeru. So for those of you who are participating on the webinar, if you do have questions, please submit those using the chat box feature to the corner of your computer screen. I do have a couple questions. The first one is for you, Neeru. This question is, what is the relationship between HPTN and other networks such as the HVTN? And how do studies potentially collaborate and communicate, and findings inform across these networks?
Thank you. That's a really great question. So One of the new innovations as a prevention intervention is the use of monoclonal antibodies for prevention. And this approach actually really can inform vaccine research as well as can serve as a new prevention modality. And therefore, it really is a very unique opportunity to collaborate with a network that has expertise as well as conducts a lot of vaccine research, which is the HIV Vaccine Trials Network, and expertise and conducts systemic and other prevention modalities, which is HIV Prevention Trials Network. And so the leaders of both these networks saw an opportunity to collaborate, Dr. Mike Cohen and Dr. El Sadr, as well as Dr. Larry Corey and Dr. Scott Hammer, and their leaders. And this was obviously very much supported by the Division of AIDS and National Institute of Allergy and Infectious Diseases. So when the Vaccine Research Center at the NIH identified a product that could be evaluated as an antibody-mediated prevention, and hence the acronym AMP, it was an obviously good way to collaborate between two networks. And so the leaders got together, identified and collected and identified team members that represented both networks, and in a very collaborative way, identified sites as well from both networks. And that has resulted in fruitful collaboration of two studies, HPTN 703, HPTN 081, and HPTN 704, HPTN 085. Together, we call both those studies fondly as AMP studies. And then, because that was our first generation in this new modality, and there is promise of optimizing new monoclonal antibodies as well as combining new monoclonal antibodies, other studies along that path have been conceived and are being developed in collaboration with that network.
Okay. And can I just add, Jonathan, one other comment? In terms of the finance program and collaboration, in particular in support of the AMP trials, you know, both the PTN and VTN have actually tried to standardize the template and the funding approach for the benefit of the sites, recognizing that, you know, that multiple networks could increase the administrative burden at sites. So on purpose, we've tried to standardize so that that burden is minimized. And we've done that, you know, initially through a face-to-face meeting, talking about the approach and the template to be used. And then we've had follow-up phone calls, again, as check-in points or at key times when deliverables are needed, again, for the benefit of the site.
Thank you, Kathy. Thank you, Dr. Sista. The next question looks like it's for you again, Niru. Can you share some examples of structural intervention research questions that the network has conducted or is conducting?
Sure. And again, thank you very much for that question. I was hoping people would ask so that we could explain. We have evaluated structural interventions even in phase 3 studies. And I'll give you an example of a phase 3 study conducted in South Africa. It was looking at conditional cash transfer to keep young women in school based on school attendance and evaluating the effect of that on HIV incidence in that population. That study, the number is HPTN 068. It was chaired by by Audrey Pettifer, who's also the chair of our Adolescents at Risk Committee. Again, really, this was one of the largest studies that recruited young women, young adolescents, starting from the 7th or the 8th grade through high school, graduation of 12th grade, young girls in schools, 2 arms, and one arm had conditional cash transfer based on school attendance, and the other arm had standard of care sort of counseling, but not that financial incentive. And that study was completed and has been published. The study did not show impact on HIV incidence, but I would like to note primarily because the attendance in both arms was actually very high. And so I won't say primarily because, but it was certainly a contributing factor because we did not see a difference in attendance between two arms. Once we go into a school community or school communities with interventions, as you all can perhaps understand, in general the engagement in schools increases, and young girls responded to that by increasing their attendance at school as well in that study. But there is a lot of data that has come out of that study in addition to just the impact on HIV incidence. It continues to get presented and published, presented at seminars and conferences, as well as published in peer-reviewed journals, and is probably a very rich source of data on adolescent girls.
Thank you, Niru. We have a question from the Spanish-speaking line. The question is, the sites that do not belong to the clinical trials unit and are contracted by project, can individuals at those sites access the scholarship program?
So the Scholars Program is for a scholar, and they will get mentored by a mentor at some HPTN site. So yes is the short answer. It is not necessarily only for scholars at sites. It is for young investigators interested in HIV prevention that would like to participate in HPTN research agenda.
So a follow-up question to that, Niru, is can pharmacists, so if I'm a trained pharmacist, could I apply for the Scholars Program, or is it just like MD, PhD?
So I would really encourage everyone to go and look at the actual Scholars Program on the website. To be able to apply for the program, one has to propose research that gets then vetted with a mentor, and when it's reviewed, it is the research concept as well as whether the feasibility of that research concept, either with using HPTN data from older completed studies or whatever the concept may be. So I wouldn't want to say no if there's an interesting idea that a pharmacist has using HPTN data or availability of some sort of HPTN information, and that a mentor is willing to mentor about, it can certainly be considered.
Can I, Jonathan, this is Kathy, can I just add, and again, Niru has kind of described the Scholars Program and mentioned that there is on the website, there's additional information under the research category, including deadlines that are coming up for both the domestic and the internationals program. And so I think we can highlight those today, one being the January 5th for the domestic program and January 29th for the international. So I do encourage people to really look at that description, and if there are questions, there's a point of contact listed on the website. But there is additional information for people who are very interested in this program that, as Niru has said, has not only been successful, but it has been funded well thanks to those people who have gone through the program.
So Kathy, thank you for that. The next question is for you.
Okay.
How does a site that is selected as a protocol-specific site become an HPTN affiliated site? I believe we might refer to that as core funding sometimes.
Yeah, and I'll address it briefly and also let Niru address it from the network evaluation perspective. But those that are protocol-specific remain protocol specific until kind of deemed by both the Network Evaluation Committee leadership and a consultative process with NIH to actually receive, to one, be affiliated as a HPTN site, which equates to basically additional infrastructure core funding. And there is a process by which NIH actually goes through to affiliate sites to an HPTN. And Neera, I don't know if there's anything else you want to add to that.
Yeah, so we go outside of our HPTN sites, as I mentioned, when we have the need for a specific protocol. So often those protocol-specific sites are only participating in one protocol. If there is no other need for that site, then it becomes, it remains a protocol-specific site. If, however, we see that the same site, because of either access to the population or their study conduct or how well they've done, and as Kathy mentioned, evaluations, continue to be recruited as protocol-specific sites in other studies in HPTN, then the Performance Evaluation Committee, the HPTN leadership, and as well as the Division of AIDS, in a consultative process, decide that it would be best for them to be converted to a HPTN-specific site.
Thank you, Neera and Kathy. This next question is speaking about collaboration among or between networks, such as the collaboration that the HPTN currently has with the HPTN in conducting the AMP study. This question says, how is what we learn from one network, such as relationships with target populations and research capacity, how does that inform how this knowledge is applied across other networks? So what we learn in the PTN, how is what we learn applied in some of the other, our brother and sister networks?
Absolutely, and thank you again for the question. I think, first of all network leaders meet together on a regular basis on a conference call conducted by, through a cross-network collaborative group. So they're well aware of each other's protocols and so know what they learn from each other's protocols. They're also, all of them, experts in HIV prevention, so they're also aware of the scientific papers and literature that comes out of each other's network. But more specifically, for example, we conducted a feasibility study trying to recruit and retain MSM, the men who have sex with men in Africa. This was an HPTN study, HPTN 075, and obviously that study was successfully completed. We could recruit and retain the number of men we wanted to in that study, but that allowed then that information to be not only shared but taken advantage of by other networks. And I understand that the Microbicides Trials Network, for example, is going to go to the same sites and also learn lessons learned from our study to be able for them to enroll participants in their study in Africa. And that's just one example where HPTN's research and site shared lessons learned were shared with other networks. Similarly, there are other lessons learned from HPTN or MTN or Impact that HPTN benefits from. So it's a very, again, a bidirectional collaborative process.
Thank you, Neera. With that, we've run out of time, so I wanted to provide just a little bit of space for Dr. Sista and then also Mrs. Henson just to provide some closing remarks.
Thanks, Jonathan. Again, I would like to welcome new sites and thank all of you for spending the time to learn more about HPTN. It has a very vibrant scientific agenda. We did not get into the more specifics of the scientific agenda this time, but there will be plenty of opportunities for all of you to learn about the many diverse type of studies that are ongoing in HPTN, very seminal studies evaluating new interventions. I'll again like to highlight that we are meeting together at the regional meeting in Johannesburg, South Africa, where you will learn even more about the scientific agenda at the plenary sessions. I once again would like to really thank you and welcome aboard to the HPTN.
And to echo Niru, again, our appreciation for all of you for not only participating in today's call, but for what you do on a daily basis to help end the epidemic. A special thanks to our colleagues whereby English is not a first language. And so, we've been fortunate enough to have our translators, Donati translators, help us today in multiple languages to carry out the message about what we're doing as a network. And again, my thanks to the community who's also on the call, because I know they're charged to do other activities, but you took time out of being in the field to be and hear more about the network. So my thanks to all of you. Again, we weren't able to answer all the questions, and we recognize that. We will try to address them on email, and I encourage people to continue to ask us questions because we were somewhat general today, but we welcome any specific questions offline. So again, my thanks to all of you for participating. And Jonathan and Nakia for your help, as always, in setting this up.
Thank you, Kathy. So as a follow-up to Kathy's message, if you do have additional questions, if you could please send those to me. My email address is jlucas@shi360.org, and I'll make sure that your questions get answered. As a reminder, the second webinar in this series is scheduled for October the 11th. It's going to provide an overview of the HPTN Statistical and Data Management Center and then also the HPTN Laboratory Center. We're also going to be posting these recordings again on the HPTN website, www.hptn.org, and it will be available in English, Spanish, Portuguese, Vietnamese, and Thai, so that if you'd like to use these in the future with staff, orientation of staff, you can do that. Please also take the time to complete the evaluation that will be sent out by Nakia at the end of the webinar, because we will use that information to help inform webinars that we have in the future. Again, thank you for your participation, and I hope this was helpful to you all. Have a good day or evening.